Well, I'm thrilled to be here today to talk to all of you about seeking life's polymers at ocean worlds. And I'm going to start today with my bias, which is life as we know it. It's the only example of life that we've got so far. And so when I think about life, you can think about all the different physical components of life and searching for those. And we also might think about three different classes of components. For example, the proteins, which are polymers, they're polypeptides, sequences of amino acids that fold into structures that are the machinery, the metabolic machinery of life. We can also think about informational molecules like DNA or RNA, which are polyelectrolytes, also polymers with a charged backbone that separates their physical and chemical properties from their informational properties, which is really important in order to enable copying. And then finally, we can think of a third class of molecules compartment forming, for example, lipids that make up cell membranes. There's also other kinds of molecules that might help form compartments, sugars, for example, but lipids is a very common one. And so I come to the task of life detection with this bias, and I've spent most of my astrobiology part of my career looking for life as we know it. But when we think about going to ocean worlds, we're now talking about going to places where, unlike, for example, Mars, which has experienced a lot of meteoritic transfer between Earth and Mars due to large meteorite impacts, ocean worlds like Europa, Titan, Enceladus, other places in our solar system have not experienced that meteoritic transfer, and the potential for life hitching a ride on a space rock is much greatly reduced to the point where if we're looking for life, it's likely to be life as we don't know it. And we can't make the assumptions that we might make about the kinds of the specific molecules that we might want to look for. So we could also target properties of life. But anyway, we're going to be going to some of these places. For example, soon with Europa Clipper, maybe a lander in the future, a dragonfly at Titan, but we don't have a candidate mission going to Enceladus quite yet, although we're hearing about some very strong possibilities for the future. And before I get to that, I'd just like to say what makes Enceladus so special in my view. And I think there's probably a good amount of agreement on this, but number one is the plume. It provides access to the subsurface ocean that we think is there from orbit or from the surface, where we can capture falling plume particles or maybe even venture into one of the vents. And the other thing is we already know that there are organics there. Studies have shown that there are high molecular weight organics present in the plume particles as measured by Cassini. And there's a plausible case for habitability. We know that the elements required for life are present. We know there's energy due to the higher temperatures of the vents, the flexing of the icy shell. And we think there are probably vents in the ocean as well at the rock-water interface, which could be an analog for proposed vent origins of life. It's also a relatively mild radiation environment in comparison to Europa, for example. And due to its orbital fluctuations, these vents may actually be somewhat short-lived on the order of, say, millions or tens of millions of years. And if that's true, and there's life there, that would imply that life is common in the universe, that it can evolve quickly, for example. So this is a really interesting test case for astrobiology. Of course, it's also hard to get to. The polar orbit is unstable. We don't know the gravity field, so planning a mission is tough at this point, and you're not going to get a lot of sample from orbit, but these are challenges that we can work. So I think in this forum previously, you may have had a pretty good summary of the Orbilander mission concept. And this is just a very quick snapshot that I will use to refer you to an amazing mission concept study that was led by Shannon McKenzie for the decadal survey. And the very brief summary that I will give is that this mission would both be an orbiter and for about 1.5 years, and then would go to the surface and then collect additional sample using a kind of funnel-like structure that you can see on the right there. And if you go read that report or you go read the associated article in Planetary Science Journal, you can review the science objectives. And one of those is Enceladus inhabited? And so 3 aspects that I'm going to focus on today is detecting polyelectrolytes, which the study proposed to do using nanopore technology. The second is functional groups of organic molecules. And the third is amino acid abundance distribution, looking at the abundance of different kinds of amino acids. And typically this is done looking at the most simple to the most complex, where we would expect there to be, if there wasn't life, the complex amino acids are much more unlikely to be made. So when I think about this, again, going to life as we know it, this is a representation of the scale of life from things that are tens of microns, and we might see sailing around in the air, like pine pollen or yeast or dust, large dust particles, or to single cells at the micron scale, COVID, as we've come to know and not love, or smaller subcellular entities like ribosomes, about 20 nanometers across, or a DNA double helix, or single strands of a polypeptide that folds into a protein. And so we have to ask ourselves, what are some methods for sensitive, specific, and agnostic detection? And so I'm going to try to address that question, focusing on nanopore and a related technology, nanogap, today. So for each of these classes of molecules for life as we know it, we can think about what would be our approach to detect life as we don't know it. And so one of those that I alluded to a moment ago is the amino acid abundance distribution, where if we have amino acids of different complexities, say from small to large, or number of carbon atoms, or some other measure, which I'll talk about a little later, if there's no life present, we would expect the more complex ones to be very low in abundance. And whereas life uses a wide range of different amino acids, and so we expect a more, let's say, somewhat even distribution. And another molecule of key interest would be polyelectrolytes. And there we can think about, instead of the nucleic acids DNA and RNA in all known life, we could think about maybe their variations. Maybe they're TNA or GNA, or maybe they're a different polymer. But it's been argued that for aqueous-based life, polymers with a repeated charge on the backbone might be universal. And the reason is that helps separate the copying from the information. So if the backbone dominates the properties of the molecule, that means that if you change the information, you're not changing the properties of the molecule. And that's really important, because if you change the properties of the molecule when you change the information, that's going to make evolution really hard. It's going to interfere with copying, for example. Now, the existence of informational polymers invokes the idea of coding. And that means there might be a translation apparatus, something that translates the informational molecules into the metabolic ones. And so we're going to talk about that as well. I won't focus on the compartment forming apparatus today, like lipid molecules, for example, but that's another area where we could probably come up with some universal biosignatures. For example, biology adding chunks to molecules, like a certain number of carbon atoms. So I'm going to talk about these 3 technologies: SETG, the search for extraterrestrial genomes, where we're using biological nanopore sequencing and trying to embed that in an instrument. The second is solid state nanopores. And the third is nanogaps. And I'll explain each of these in turn, where we're developing an early stage instrument called the Electronic Life Detection Instrument for Enceladus Europa, or ELI. Yes, I like contact. So strand sequencing works by taking a nucleic acid, typically DNA, but also RNA, and going through a bunch of steps to make a double-stranded molecule, which can be unzipped by a motor protein and put through a biological pore. And this pore creates a channel in a high resistance membrane and allows the ionic current flow through that pore to be measured. And the motor protein is really important. It slows the DNA down, and it's what allows us to measure the ionic current in a meaningful way. And we get a squiggle, which we can then convert the changes in ionic current to infer the sequence of bases. And this has really grown massively over the last decade. And I want to share with you just a few of the selected accomplishments from our group. There have been many from many other groups as well. For example, Aaron Burton and Sarah Wallace doing the first sequencing in space, as well as I think Catherine Maggiore is on the call today and has been involved in some nanopore sequencing as well, if I remember correctly. So one item is strand sequencing, where we showed that we could use this to detect inosine, an astrobiologically relevant non-standard base. Another thing that we did was we wanted to establish that the false positive rate for this technology was low. So we literally sequenced nothing in as many different ways as we could think of. And through that work and through testing different algorithms, we were able to show that despite some original early false positives, those have since been addressed through changes in chemistry and base calling algorithms. We also did a sequencing in a thermal vacuum chamber under Mars-like conditions, so we could understand what we need to do to control the environment and ensure that we can operate devices at typical at permissive conditions for nanopore sequencing. We also did low input sequencing, demonstrating we could detect Bacillus subtilis DNA from about 2 picograms of input, which represents, in our particular experimental setup, a sensitivity of about 1 part per billion. We also did a parabolic flight where we demonstrated by looking at the ionic currents and very specific signals on the time scale of 1 base, that this system is extremely robust to vibration and G level. So this bodes well, and we did this at Mars and Europa and weightless conditions. So I'd like to move on for there. Hopefully in the discussion, we'll have the ability to discuss what are some of the challenges that remain for this technology. I'm going to move on to solid state nanopore for now. And there we've used to date the most data that we have so far is from this device, the Ontera Nano Counter. This is a commercial product. It's very low power. It uses these little tiny chips, and within the very center there is a nanopore. You can adjust the size through electrical conditioning between about 10 and 40 nanometers. And because it uses a high salt buffer, it's salt tolerant, which is important for ocean worlds. You can detect a variety of different analytes. There's very minimal sample preparation, usually just a concentration if you want to concentrate your analyte of interest. And then the analyte passes through a nanopore, a small synthetic pore, and the ionic current is again monitored. And so some target applications would be measuring the length of polymers or looking at other subcellular structures. So Jordan McCaig in my group has been, she's a doctoral student in Earth Atmospheric Sciences, and she has an upcoming presentation where you can learn more about this at the Ancient and Future Brines Conference. And I want to share just what does an actual event of a molecule look like. So here's an example of DNA going through the pore. So we can see that the ionic current drops, holds steady at about minus 100 picoamps due to the blockage of the DNA, and then returns to its normal baseline. We've subtracted the baseline out for the purposes of this illustration. And then with a plasmid DNA, you can see that the ionic current drops about twice as much. And you can also measure the length of the DNA, at least very approximately. So while the red line is one event from a 1 kilobase ladder DNA, the black line is a high range DNA. So you can measure length, and we're working on quantifying how accurately you can do that. So you can do this for all kinds of different DNA molecules. You can map out how long the DNA stays in the pore. So you can see on the right-hand side here, this ladder is staying in the pore at different times, but it has a similar drop in current measured as a change in the conductance on the vertical axis. And you can see the plasmid has a different change in the conductance and generally goes through very quickly. And we've also done this with single-stranded RNA and double-stranded RNA. We've also done this with ribosomes. And so this scale here is very different. So there's a much higher delta conductance, and we interpret this upper peak as intact ribosomes, and some of the other, this other peak down here and some of the other events as ribosomal unfolding in the buffer that we're running, which is lithium chloride. And so we think that this technique could be used to detect a translational apparatus that's not a ribosome in addition to ribosomes. So stay tuned for more on that. Now, there are some challenges with solid-state nanopores. They're really hard to make right now. There are some new techniques that are making it easier to make relatively small nanopores. But one of the big challenges when you make a small nanopore, when you're monitoring the current, is the DNA or some other molecule zips through it extremely fast. So this drives you to need extremely high bandwidth, like 5 or 10 megahertz. And we're still not able to get down to single base accuracy because that happens just so quickly, and you need a very high bandwidth. So what we're doing, while I fully believe that this is possible to get to single base resolution, it's really marginal and it's sensitive to noise. And so we're taking another approach in parallel, which is called the nanogap. And a nanogap works by, in this case, it's a gold nanowire on a silicon chip. We put it in bending through an actuator, and this causes bending, thinning, and breaking of a gold nanowire and the formation of an atomically sharp junction. And by applying a voltage here, we can monitor the tunneling current across that junction, and it dramatically increases when there's the presence of a small molecule, which can effectively decrease the gap width. And so here we think we can use this technology to measure an amino acid abundance distribution, as well as charged linear informational polymers, targeting prebiotic, ancient, or extant life. And one advantage here is that the bandwidth requirements are 100 to 1,000 times lower than solid-state nanopore. And Jose has been working on this and we've released a preprint of some initial work with our very first prototype. And so the upper left here is the original system that was developed by Osaka University and Masateru Taniguchi's lab. On the upper right is our first prototype. And on the lower left, you can see inside the Faraday cage where we had literally a manual micrometer where we would adjust the bending of that chip. And this is not a great system, but it was what we could build quickly and efficiently and get it and demonstrate that we could do it in a small format. And this was the best data that we obtained with this system. And I say was because we've now moved on to automatic gap control with piezos. And this is going to allow us to get much more detailed data. So this is kind of our first light experiment with 10 micromolar L-proline. And you can see the example on the right of characterizing a couple of these events. We can characterize the dwell time and the tunneling current. And we can also apply machine learning approaches to looking at different amino acids. And this would allow us to characterize the conductance of different amino acids, which our collaborators have already done. And so we used that and we looked at when these different amino acids were fixed into the genetic code. And what we found is that the HOMO-LUMO gap, which is a measure of chemical reactivity or ability to pass electrons, declined. So basically, meteoritic amino acids are insulators, but genetic or late appearing amino acids in the genetic code are semiconductors. They're better at passing electrons. So indeed, what we also found is that those low HOMO-LUMO gap amino acids have higher conductance. So this means that the nanogap is basically, we're going to get the best signals for the amino acids that are biogenic. So This led to us proposing as a defining property of life, improved electron passing, or life as a semiconductor, where life needs to control the flow of electrons. So I think, by the way, in addition to this, we can detect polypeptides and polyelectrolytes. And our colleagues have done a great job over the last decade demonstrating that this can be done. And this is an example from a 2012 paper by Takahito Oshiro, looking at RNA and showing that the nanogap conductance signals allow distinguishing different bases and also obtaining short subsequences, which we expect can be improved in the future with better molecular control. So we're currently trying to assemble these different technologies into one system that we envision called single molecule analysis of life in liquid. And what we anticipate is being able to analyze cells or particles, lyse them, and do an initial screen with a solid state nanopore, where we would hope to identify potential informational polymers or perhaps a translational apparatus. And then, if warranted, that same sample could be passed on to a biological nanopore sequencing device to identify any forward contamination or life as we know it, or to a nanogap-based device to seek evidence of life as we know it or don't know it. So all three of these technologies would allow us to detect polyelectrolytes. And in addition, the nanogap technology would allow us to detect an amino acid abundance distribution or other functional groups, at least as measured by their ability to enhance tunneling. So thank you very much. That's where I'm going to leave things for today. And I really appreciate the opportunity to come talk to you. I'm grateful to all of the members of my group and also to NASA and NSF for support. Thank you so much.
That was excellent. Bunch of virtual clapping.